Asexual reproduction in protozoa creates clones. But let's say there is a sudden change in temperature or pH in the external environment. What do you think would happen to these clones? Well, all of them would die because there are no genetic differences between them. If the change affects one amoeba, it's going to affect the other. And that could be a problem if they want to maintain their population. We also discussed about the division of Paramecium, where one nucleus divides by mitosis and the other by amitosis, and yet they are clones. In spite of the difference in cell division, we only consider the mitotic division of micronucleus to say that the daughter cells are clones. So here too, like we discussed in amoeba, the cells are going to die when there is some sort of an environmental change. In addition to that, the macronucleus of the paramecium is a ticking bomb. The amitosis is going to build up chromosomal imbalances, and that can affect the functioning of the cell. Therefore, in order to prevent themselves from dying out and to better survive in an environment, the protozoans reproduce by sexual reproduction in regular or irregular intervals in between their asexual reproduction. This is the type of reproduction where two matured individuals produce offsprings. And here maturity means sexual maturity. When organisms attain sexual maturity, they produce gametes by the process of meiosis. And because they are produced by meiosis, the gametes are going to be haploid. The process involves a fusion of a male and a female gamete. Both these gametes are haploid. When they fuse together, they give rise to a zygote, which is diploid. Unlike asexual reproduction, the offsprings in sexual reproductions are not identical to the parent, nor are they identical amongst each other. So we know meiosis gives rise to haploid gametes, right? So during the process of meiosis, we see a phenomenon called as crossing over where genes on homologous chromosomes are exchanged. This leads to genetic recombination. This is the first level where we see such kind of recombinations being introduced. Two completely different individuals come together and there is a random fusion of their gametes. This also leads to genetic recombination, and this is the second instance where such a genetic variation is introduced. From this, we can conclude that sexual reproduction introduces genetic variations where asexual reproduction do not. Sexual reproduction occurs through the process of syngamy. This is the fusion of the male and the female gamete. There are different types of syngamy based upon how the gametes appear. For example, in isogamy, we see the fusion of morphologically similar gametes, meaning they look similar and we are unable to differentiate which is the male and female just by looking at them. This is seen in monocystis. There is also anisogamy, which is the fusion of morphologically as well as behaviorally dissimilar gametes. Male gametes here are usually smaller and motile. The female gametes are larger and non-motile. This is seen in Plasmodium. There is also another unique type of syngamy called as hologamy. Here gametes are not formed, but two premature protozoan individuals fuse to form a zygote. This is seen in Trichonympha. During the fusion of the gametes, there is a stage where the cells are fused, but the pronuclei of the gametes are still unfused. Later on, the pronuclei also fuse to give rise to the zygote. The process of fusion of the pronuclei is called amphimixis, and the resulting nucleus is called as a synkaryon. Another phenomenon we notice is the conjugation. It is not a true sexual reproduction, but it could be considered as part of sexual reproduction, and it is not used specifically as a mode of multiplication. But because it involves fusion of nuclei, we consider it to be part of sexual reproduction. Adverse environmental conditions can trigger conjugation, and this is seen in ciliates like Paramecium and Vorticella. It is the temporary union between true senile ciliates. Senile means older organisms that have undergone multiple rounds of binary fission. Conjugation cannot occur randomly between two ciliates. They must belong to two different mating types or clones, but to the same species. Here, mating types refer to the genetic arrangement within these organisms. It is observed that Paramecium usually shows two types of genetic rearrangement, the A and the O. A undergoes binary fission to give organisms with A mating type, whereas O undergoes binary fission to give mating types O. Two organisms that are A or O cannot undergo conjugation, but an A organism and O organism can conjugate. This ensures that there is exchange of genetic material, and this helps in creating genetic variation. Okay, now let's quickly see the process of conjugation. So when two compatible cells meet, they align with each other in a side-by-side fashion. They become partially fused by protoplasmic bridge. At this point, both the cells have a diploid micronuclei. The micronuclei undergo meiosis and produces four micronuclei in each of the cells. The micronuclei are haploid. Out of the four micronuclei, three micronuclei disintegrate and only one remain. The remaining micronuclei divide by mitosis. This results in two micronuclei in each of the cells. The mating cells seem to exchange one micronuclei amongst themselves, after which the cells go in their separate ways. The micronuclei in each of the cells fuse and become a diploid micronuclei. This is what we discussed as syngamy earlier. The diploid micronucleus undergoes 3 rounds of mitosis to give rise to 8 micronuclei. The original macronucleus now starts to disappear. Out of the 8 micronuclei, 4 of them become new micronuclei and 4 of them become new macronuclei. The cell then begins to divide and gives rise to 4 cells, each with their own new micro and macronucleus. So, if this is not an actual method of reproduction, why do these cells undergo conjugation? The first main reason is that this process introduces genetic variation. Genetic variation ensures that when there is a drastic environmental change, at least some of the daughter cells are able to survive. These cells usually divide by binary fission, right? But multiple rounds of binary fission can lead to genetic imbalance, especially in macronucleus, which divides by amitosis. So, when we introduce a round of conjugation, we can restore the vigor and vitality of the cell. Finally, the macronucleus is involved in the cell metabolism. When the genetic balance is restored in the macronucleus, it helps in accelerating metabolism.